So thank you for attending today's webcast, Latest Advances in Cancer Research. My name is Nicole, and I'm the moderator for our online community, CancerConnection.ca. Before we begin, I wanted to cover a few housekeeping items with you. The screen you're seeing right now is completely customizable. You can move around the pages and make the screen larger by selecting the Maximize icon. Near the bottom of your screen, there are several buttons. I'll quickly go over those, moving from left to right. The first button is a question mark, and this is our help guide if you have any technical problems with seeing or hearing the webcast. The next one is your volume control, and then a button that controls your view of the slides. So you'll want to keep this one maximized. To the right, you will see a Q&A button. We're really looking forward to hearing from you, so if you have questions for our presenters, you can send them in by typing in the box and hitting submit. We'll answer these questions live at the end of the webcast, and you can send your questions throughout. Please note that our presenters will not be answering personal medical questions. Next is the sharing button. We hope you will share this webcast with others in your social network, those who might also be interested in this topic. Following that, we have a button for resources, where you will find a copy of our clinical trials brochures. The next one is a bio of today's presenters. You can select this widget to learn a bit more about them. Next is the link to our online community, CancerConnection.ca. And finally, on the very far right is a survey for you to provide feedback about today's webcast. I hope you'll take a moment to let us know what you think. Your input helps us to plan all of our future webcasts. Tonight, as you can see, we have three researchers, Natalia, Matt, and Sue. And at this time, I would like to introduce you to Natalia, who will begin presenting the presentation.
Okay. Hi, thank you for joining us for today's webcast. We'll be talking about the latest advances in cancer research with an emphasis on some new advances in treatment. And before we begin, wanted to give you a little bit of information about ourselves. So we are young scientists who volunteer with the Canadian Cancer Society, and our volunteer group is called RIOT, or a Research Information Outreach Team. And our goal is to educate the general public about cancer and its research with an emphasis on the progress and promise of cancer research. And if you want to learn more about us or read interesting articles on some of the latest advances in cancer research, please check out our website at torontoriot.com. Okay, so, sorry. I know that each and every one of us has been affected by cancer in some way or another. And sometimes it feels like there isn't much progress being done. But we are here to tell you that we are making progress. And our knowledge of cancer and just how complex it really is expanding every day as researchers study this disease. Now, our progress is in large part due to the advances in technology that scientists use to study this disease. And we're using that knowledge to bring forth advances in cancer screening and treatments and even create innovative treatment strategies. Okay, so when we talk about standard cancer treatments like surgery, radiation, or chemotherapy, it sounds like not a lot has changed over the years. But the truth is that these treatments have improved immensely. And to give you an analogy, think of telephones. Think about how much the telephone has advanced since it was first invented in the 1800s. Nowadays, phones are mobile and they act as mini cameras and computers all rolled into one. And they're still phones and they're still used to communicate. And the same could be said for these cancer therapies. So to give you an example, in the 1950s, the Canadian Cancer Society funded researchers that developed the Cobalt-60 unit. Cobalt-60 was one of the original units that was used to deliver radiation to treat cancer. And this discovery paved the way for modern-day radiation. Today, researchers are still continuing to find ways to improve radiation, including better imaging and better ways of delivering high doses of radiation that are improving the survival of our patients. And much like the evolution of the telephone, there have been significant changes in how we treat cancer using traditional techniques such as radiation and chemotherapy. But in addition to that, it is our basic research that is carried around the world that is helping us to bring forth new ways to treat cancer. So what is basic research? Well, what basic research means is that scientists are using cancer cells grown in a petri dish or model organisms like fruit flies, zebrafish, or other animals like mice to help answer questions about cancer. How does cancer start? How does it spread? What drugs stop cancer growth? Or how do cancer cells become resistant to those drugs? Those are just a few of the questions we try to answer with basic research. And so as you can see, it's extremely important because we have to understand how this complex disease works in order to find ways to treat it. And basic research is why scientists are now trying a new way to treat cancer called personalized medicine or targeted therapy. And so that's what I want to talk to you about right now. So personalized medicine or targeted therapy means finding the right drug for the right patient. It means tailoring the medical treatments that are available to the individual characteristics of patients, not developing brand new drugs for each individual patient. In the past decade, we've seen a major transition in drug therapy for cancer. There's been a shift from the broad spectrum cytotoxic agents like chemotherapy to highly targeted therapies. And we're seeing this for kidney cancer, malignant melanoma, non-small cell lung cancer, breast cancer, and chronic myeloid leukemia. So instead of treating everyone the same, with the same diagnosis and same treatment, we can look at the genetic makeup of the patient's tumors and find out whether they carry a specific mutation, and then provide the patients who do have this mutation with a certain drug, which targets that mutation. So hopefully the patients that have the mutation will benefit, and the patients that don't can be spared the expense and the side effects of these drugs. I will outline some of the advances that have been made, but also present some of the challenges that are associated with this cancer treatment strategy. So here's one example of targeted therapy. So chronic myeloid leukemia is a cancer of the white blood cells. And in the 1960s in Philadelphia, scientists found that the chromosomes, which are these finger-like structures that contain a person's DNA, were normal. A piece of chromosome 22 was missing, and chromosome 9 was longer. And this is called a translocation, and it appeared as though this translocation was the cause of CML. Then about 13 years later, scientists figured out what two genes had been fused together in this translocation. They were called BCR and ABL, and this fusion caused cancer. So for the next 20 plus years, researchers developed drugs to treat this BCR-ABL translocation in order to treat CML. And in the early 90s, a drug called Gleevec was created. And when it was tested in patients, 100% of the patients in a phase 1 study, which is used to test safety, responded to the drug. This was unheard of. A 20% success rate is usually positive. Further results in clinical trials were very successful, and Gleevec was approved in 2001. The results even made the cover of Time magazine. It was the first time scientists were able to understand what caused a particular type of cancer and make a drug to treat it. So what is the result of Gleevec? Cancer is controlled by taking one pill a day with virtually no side effect. And pre-Gleevec, the survival of CML was less than 20%, but now it's almost at 90%. And this definitely highlights the significance and impact that this drug has had. Another example is a drug called Herceptin. So back in the 1980s, researchers learned that a gene called HER2 was amplified in breast cancer. And those that had more HER2 caused cancer cells to grow very quickly, and these tumors were very, very aggressive. In the 1990s, researchers found a drug to target HER2. It was called Herceptin, and it has been very successful in treating breast cancer patients whose tumors overexpress this protein. Now we know that HER2 is amplified in a subset of gastric cancers, non-small cell lung cancers, and colorectal cancers. So the question remains, can drugs like Herceptin be used to treat these cancers too? And as technology for science improves, we are sometimes able to move biological findings from the lab into the clinic much faster. Technology is helping scientists identify new genes and mutations driving cancer so we can create drugs to treat them. And an example of this is a drug called crizotinib. In 2007, scientists identified two genes that had fused together, much like PCR and ABL, but these were called EML4 and ALK. This fusion protein drove the growth of non-small cell lung cancers in a small percentage of patients. And in 2010, it was discovered that patients that had this mutation responded very well to drugs that treated ALK. Within a year, the drug was approved by the FDA to treat lung cancer patients whose tumors contained this fusion protein. So it took us over 40 years to learn about the genetics of CML and find a drug to treat it. But now with the advances in technology and a greater knowledge of cancer, we were able to bring forth a new treatment in 4 years. Not every treatment will move to this clinic, to this pace, obviously, but it is quite an impressive achievement. However, as I mentioned before, cancer is very complex disease. And even with the last example that I mentioned, we are seeing lung cancers that are becoming resistant to crizotinib due to the secondary mutations in ALK. This may happen to many drugs as well. And as a result, we are working very hard on developing newer and better drugs. So for instance, a new drug called certinib, which is a newer generation drug targeting ALK, was approved in 2011 and is showing promising results for non-small cell lung cancer patients who have developed resistance to the original crizotinib medication. In addition, similar to what's being done for diseases such as AIDS, researchers are looking at using combination therapy to fight resistance. So genetic tools are being created to identify the most powerful drug combinations to use in patients instead of just guessing which combinations to use from the hundreds of available cancer drugs. And there are other challenges as well. So these drugs are not less toxic than chemotherapy, and so toxicity and side effects from targeted therapy are also things that will have to be dealt with as these drugs come about. And in addition, we need to think about the cost and resources. Can smaller cancer centers undertake this approach, where diagnostics and treatments are more focused on genomics, and innovations in medical drug development and clinical testing may lower costs? So for example, instead of a large unselected patient population for clinical trials, where 9 out of 10 drugs fail along the way, we're starting to see stratification of patients so that smaller clinical trials with higher success rates are starting to happen. And another note is that not all mutations that we see in cancer are currently targeted by drugs. Researchers are continuing to develop new drugs, but this may take time. So even though we're learning more about the mutations driving tumor growth, we still need to develop proper therapies to treat them. And personalized medicine does hold a lot of potential, and it will be interesting to see what happens in the next few years. Just as scaling a mountain takes a long time and a lot of effort, researchers are continuing to work hard to tackle new ways to treat cancer. And one idea is to combine personalized medicine with new approaches like immunotherapy. So now Matt will tell you more about immunotherapy.
Hi everybody, my name is Matt Hall, and I'm going to tell you about the exciting world of immunotherapy. I'm going to start by explaining what immunotherapy is. Immunotherapy takes advantage of the body's natural defense system, the immune system, in order to target cancer cells and kill them. You look at the image on the right, that is an image of a T cell. A T cell is one of many types of immune cell that work to defend your body from foreign invaders. These foreign invaders can be bacteria or viruses. A big question in research is, can T cells kill cancer cells in the same way that T cells kill bacteria and other foreign invaders? Before I get into that question, I want to address how it is that T cells recognize foreign invaders. Every cell is encased in a membrane. And on that membrane, there are proteins which identify the cell as what it is. So in the diagram on the slide, you can see the blue cells, which let's say the blue cells are your own cells. All of the cells in your body have a particular cell surface protein which allows the immune system to recognize that these are in fact your cells. If the immune system detects your cells, it's not going to do anything because your cells are helping you to stay alive. If you look at the white cell, it is a foreign cell. It has a different set of cell surface proteins. In the diagram that I've drawn out for you, the yellow stars represent our own cell surface protein, and the green diamonds represent foreign cell surface proteins. T cells have proteins on their surface that allow them to recognize these cell surface proteins on other cells. The receptor that I've drawn is that little Y-shaped thing on the surface of the T cell. You can see that the receptor, the shape of the receptor is such that it will not fit nicely around the star protein. It will fit nicely around the diamond-shaped protein. And the two shapes fitting together is exactly how T cells recognize foreign cells. If a T cell comes into contact with these foreign cell surface proteins, it initiates a program that kills that foreign cell. This is how your body overcomes infections. The big question, as I mentioned earlier, is can this property of T cells be used to kill a cancer cell? Cancer cells, as you might imagine, are different from foreign invading bacteria. The main difference is that cancer cells come from our own cells. Cancer cells arise from mutations in our normal cells. And because of this, cancer cells share many of the cell surface proteins that are on our normal cells. However, because cancer cells are mutated, there is a theory that maybe cancer cells mutate in such a way that their cell surface proteins are changed from the normal case. This was the key question. If cancer cells mutate their cell surface proteins, it is possible that the T cells can detect these mutated cell surface proteins and treat the cancer cells with those proteins much like they would treat a foreign invading bacteria. In the early 2000s, researchers found that indeed it is possible for the immune system to protect against cancer cells. And if you look at the diagram, I've just shown you a cartoon of how this might work. So you have your own normal cells, which are the blue circles, and what researchers have found is that it's possible that through the process of turning into a cancer cell, the cell has adopted a different set of cell surface proteins. So the cancer cell on the left no longer has just the star proteins. It has developed those crescent-shaped proteins. And it's possible that the T cells can recognize those new crescent-shaped proteins and kill the cancer cells. Almost every significant research finding that affects humans was discovered through mouse research. And the same is true for immunotherapy. I'm going to briefly talk about the first experiment that really showed us that the immune system can protect against cancers. Researchers took two different mouse populations, and they were able to completely remove immune system function from one of these mouse populations. So on the top, so if you look at the pictures of the mice on the left, I've shown you a description, an illustration of how this experiment worked. So the top population of mice are the immunodeficient mice. So these mice have no immune system function. The bottom population of mice have a normally functioning immune system. Researchers subjected both populations of mice to a carcinogen, and they measured how each population of mice developed cancer. They found that in the mice that had no immune system function, there were much higher rates of cancer. And you can see the quantification of that in the graph in the middle of the screen. So the two lines represent the two populations of mice. The red line represents the immunodeficient mice that don't have an immune system, and the blue line represents the mice that had a functional immune system. And you can see that in the red population of mice, the mice that did not have an immune system, there was a much higher rate of tumor development. This was an extremely groundbreaking study. It was maybe the first time that we had conclusive evidence that the immune system is very strongly linked to the ability of organisms to get cancer. From this study, the next big question was, can the immune system fight cancer in humans as well as it does in mice? And the answer is yes. The image on the left shows immune cells targeting a tumor. The kind of bumpy region in the bottom left of that image, that's the tumor. And you can see the smaller circles kind of surrounding that tumor and attacking it. So the smaller cells are human immune cells, and they are fighting cancer. There were several. We didn't learn that the immune system can fight cancer in humans just by taking a bunch of pictures like this. There were several observations that doctors found in the clinic. One of those is that in humans, when there are a greater amount of T cells surrounding a tumor, those people had better outcomes. So if you take two patients and they both have tumors, but one patient had a lot of T cells surrounding their tumor, and the second patient did not have these T cells surrounding the tumor, the patient with the T cells, they did much better in their cancer therapy than the patient who did not have the T cells surrounding the tumor. So this was a big clue that the T cells might be fighting cancer. Another observation was researchers were able to find T cells that had the ability to target cancer cells. So much like the diagram that I showed you earlier, with the T cell receptor targeting that diamond-shaped protein, researchers were able to find actual T cells that fit into a receptor on a tumor cell. And so this was another finding that if the T cells have receptors that can locate cancer cell proteins, it's logical to assume that the T cells are fighting those tumors. And finally, much like in the mouse study that I showed you, in humans that have lower immune system function, the responses to cancer were much worse. So just like in mice, where when the immune system was functioning, there was a lower incidence of cancer. In humans, when immune system function is impaired, cancer was much, much worse. So these three findings really showed researchers that in humans, the immune system is a major player in preventing cancer. However, you might be wondering, if the immune system is so good at fighting cancer, how do people with healthy immune systems develop cancer? Well, the answer is that cancer cells can escape the immune system. And so in the diagram that I've showed you, it's basically just reiterating what I've told you before. So the normal cell will mutate into a cancer cell. And through that process, the cancer cell develops weird cell surface proteins that are recognized as foreign, and the T cell can come in, detect those foreign mutated cell surface proteins, and fight the cancer cell. However, as I've just mentioned, the cancer cell can escape this regulation. And I'm going to talk about how that occurs. The cancer cell is very tricky. It can secrete chemicals that suppress immune system functioning. T cells are subject to a wide variety of regulatory chemicals. Some chemicals stimulate the function of T cells, and some chemicals repress the function of T cells. The cancer cells can mutate in such a way that they can secrete chemicals that suppress T cell function. So in the diagram, I've represented those inhibitory chemicals as little red dots. So the little red dots get secreted from the cancer cell, and they bind to the T cell and tell the T cell, you don't need to function, nothing to see here, you need to inactivate yourself. And even though the cancer cell would be able to recognize the cancer cell, it has been suppressed by these chemicals, and thus the cancer cell will proliferate and cancer will persist. What's exciting is that we have found a way to stop the cancer cell from inhibiting T cells, and I'm going to talk about that right now. Some of you may have seen the recent PBS documentary, The Emperor of All Maladies, on cancer, the history of cancer and modern cancer treatments. In that documentary, the research of Dr. James Allison was featured, and he was a pioneer in determining how to stimulate the immune system to fight cancer. As I mentioned before, the cancer cell is secreting these inhibitory chemicals that suppress T cell function. What Dr. James Allison was able to do is introduce a drug that prevents the inhibitory chemical from affecting the T cell. So in the diagram, those little green triangles represent an immunotherapy drug. And what the immunotherapy drug does is it insulates the T cell from the action of those inhibitory chemicals being released from the cancer cell. When the immunotherapy drug is effective, the T cell is reactivated and it is now able to bind to the cancer cell receptors and kill those tumors. I'm going to share with you a couple of clinical trial results using drugs that act in the way that I've just described. One of the drugs is called ipilimumab, and in a clinical trial looking at 676 melanoma patients, researchers found that there is an increase in overall survival to 10 months from 6 months. And so the average survival of cancer patients taking this drug was increased by 4 months. It might seem like 4 months is not a lot, but in the cancer research world, 4 months is huge. 4 months is only an average, and so there are some patients who are taking this drug and have lived for years, and there are some patients where this drug was not as effective. However, this is conclusive evidence that drugs that act in this way have a very concrete effect on cancer outcomes. Another drug that is still in clinical trials is called nivolumab, and in a research study using 418 patients, they found that the 1-year survival of melanoma patients was increased to 72.9% from 42%. And so this is like a 30% increase in survival after 1 year, which is a really, really great result. So nivolumab is still in clinical trials, but ipilimumab has been recently approved by the FDA in Canada and the US, and it's being used to treat cancer, and it's becoming a routine part of the treatment for melanoma patients. And so this is really exciting, and it really, I hope it shows you that immunotherapy is a great new avenue for how we can learn how to fight cancer. I'm going to wrap up and talk about what's next for immunotherapy. The results I've just told you about are really exciting. However, they are early results. The really important findings for immunotherapy have only been effective in melanoma patients. And while this is really great when it comes to fighting melanoma, what we want to do is find ways to stimulate the immune system in other cancers as well. And there's a lot of research going into that. And yeah, people are really excited about immunotherapy, but it is still we are still in the early stages of learning how the immune system can fight cancer. And many more years of research need to be performed before we can say conclusively that immunotherapy can affect all cancers. We have really great results for melanoma, but it remains to be seen if immunotherapy will be effective in other types of cancer. So I've talked a bit about how a lot of research needs to happen, and research is really expensive. Developing drugs like ipilimumab and nivolumab takes millions and even billions of dollars. And so what Sue is going to talk about next is ways to reduce the cost of developing new cancer drugs. And the way we do that is using drugs that treat other diseases that can actually have an effect on cancer. So I'll let Sue take it away.
Thank you, Matt. So hi, everybody. Thanks for joining us tonight. My name is Sue, and I'm a cancer researcher at Princess Margaret Cancer Center in Toronto. And for the next 10-15 minutes, I would like to talk to you guys about drug repurposing. So Natalia and Matt already kind of talked about, the novel types of therapies that are coming out. And of course, the underlying theme is it's effective, but it's taking a really long time, and it's a huge financial investment. So right now I'm going to be talking about drug repurposing, and most of you, believe it or not, have probably already heard of drug repurposing. So a classic example is what I'm showing here on the left is Viagra. So in the 1980s, in the 1950s, Viagra was actually originally developed to treat hypertension, and nowadays it's predominantly used for erectile dysfunction. So when I'm talking about drug repurposing, what I'm talking about is using a drug that was intended for one use for now a novel use. So in our case, a drug that is used for disease X that now we're using to treat cancer. And another way of thinking about drug repurposing is also taking a second look at drugs that may have failed during drug development process. And I'll get into that a little bit more. But drug repurposing really plays an important role in cancer research. In fact, more than 50% of the drugs that are currently being used to treat cancer or to fight cancer are actually drugs that were originally developed for other diseases. So before I get into the wow factor of why drug repurposing is so important, I want to first go over how a drug is typically developed. There we go. All right, so this diagram shows the typical process of drug development, and it's a pretty complicated diagram, and that's because the process is quite complicated. It's a long journey for one drug to be successfully used to fight cancer. So you can see from this picture on your left-hand side, drug discovery really starts from basic research. So when I'm talking about basic research, I'm talking about research that are done in the lab, testing new drugs, or I should say drugs that were used for other indications in the fight of cancer. So we have cancer cells that we're treating drugs with, and if these cancer cells die, then that means the drug may have potential. From that point on, we then move the study into animal studies, such as mice. So now we're treating mice, tumor-bearing mice, with potential cancer drugs. And if these mice respond well, so if the tumors shrink in these mice, then it can potentially go into clinical trials. Now, when we talk about clinical trials, what we're talking about are studies done in the human population. So clinical trials involves 3 phases, phase 1, phase 2, and then phase 3. Now, the first phase of the clinical trial is typically done in healthy population. And the main goal of the phase 1 trials are to determine whether these drugs are safe to use in humans. Do they have toxic side effects? If so, then perhaps we cannot move these drugs forward. However, if they do display a good safety profile in healthy population, we can then move to phase 2. Now, phase 2, we are still trying to figure out the right concentration of drug to use, but more importantly, in phase 2, what we're trying to figure out is whether these drugs are actually effective in the patient population. So in our case, our phase 2 population would be the cancer patients. And usually the type of cancer is only a handful that we're testing. So now we're really moving down, as you can see my gradient of the arrow, from all the way to 5,000 potential treatment. Now we're getting down to eventually 1. So we just talked about phase 2 trials. Now, if it's successful in phase 2 and it's showing efficacy in terms of lowering the size of tumor and extending overall survival for cancer patients, we then can move on to phase 3. Now, phase 3 is really the phase in which it will be decided whether the cancer drug will move on to cancer patients. So phase 3 involves a huge population, usually up to 1,000 patients, if not if not more, and in multiple research sites across the world. So in this population of patients, again, now we're talking about narrowing down even further, down to treatment of one type of cancer. So in this population, it's only one type of cancer that we're treating, and if this shows effectiveness, then finally we could submit it to the government for approval. Now it might sound like, okay, they just need to put an approval stamp, kind of like what I have drawn on the slide, but it's actually a little bit more complicated because we want to really make sure that these drugs that are coming to the patients are really safe and reduce the risk of getting any other type of side effects. So once it's been submitted to the government, it takes still around 1 to 2 years before it get approved. That's if it get approved. Now if you can see on the bottom of the slide is dollar bills. And you can see that this whole drug development process can cost billions and billions of dollars. So the key 2 points that I want to mention about this slide is drug development really takes a long time, up to 15 years. And you can see from Natalia's slides that some of the drugs, such as Gleevec, actually took 40 years. So it's a really long time span, and it's a huge financial investment. And this is where really drug repurposing comes in handy. So now that you know what a conventional or traditional way of drug development is like, I want to tell you now why drug repurposing is so impactful and useful. So as I mentioned before, drug development takes a long time, 10 to 17 years, and this is really an optimistic timeline. Now the failure rate is over 90%. What that means is most of the drugs fail. They fail because of safety profile. So it's very toxic, so it won't go to the patient. Or it fails in efficacy. And when we're talking about efficacy, we're talking about whether the drug is even effective in reducing tumor burden or extending the lifespan of cancer patients. And of course, the cost is costing on average $2.6 billion. And the safety profile is usually determined, again, in phase 1, which takes time, and a little bit more tinkering in phase 2. But when you look at drug repurposing on the right column, you see that the timeline shrinks drastically, 3 to 12 years. Now, the failure is definitely less than 90% because all of the safety profiles have been worked out. So as I said, in the safety, it's already determined, and it was already determined because that drug was already developed for another disease. Now we're just plucking this drug from the treatment of this disease to cancer. So all the safety profile have been worked out. So this ultimately comes to cost. Instead of costing billions and billions of dollars like traditional drug development, drug repurposing is actually costing significantly less, on average $8.4 million. So what I'm really trying to highlight in this slide is drug repurposing is really impactful and a really great way to use a novel approach to find treatments for cancer because it's really bringing effective cancer drugs to the patients much faster. So here's an example of drug repurposing for cancer, and this is probably a drug that you guys have heard of. This is called thalidomide, and this story is really a tragic story that has ended in hope. So thalidomide was originally developed back in the 1950s and 1960s to treat nausea, to treat morning sickness in pregnant women. But it has a really bad side effect, and the side effect is children are born with deformities. So you can see on this diagram on the left side, we have a child here, and its limbs are much smaller, so there is limb development deformities. And these children can still live to adulthood, but the point is there are toxic side effects. But despite these side effects for pregnant women, what researchers have found is that thalidomide is actually quite useful in the treatment of blood cancer, and more specifically for multiple myeloma. And this is actually a pivotal finding, and it really has revolutionized the way that blood cancer is now treated, especially if multiple myeloma, because now there's second generation of drugs that are very similar to thalidomide, but work more effectively. So from this story, we now learn that a drug that was intended to use for nausea in pregnant women can now actually be effectively used in cancer treatment, and it has changed how we think about treating multiple myeloma. But this is back in the 1950s, 1960s, and we're still using that today. But where is the research going on now? Well, there's a lot of excitement around this drug called metformin, and some of you might be even on metformin. So metformin is actually the most widely prescribed drug to treat diabetic patients, so type 2 diabetes. And there's a lot of excitement around the promise of metformin because instead of treating one type of cancer, it can have potential to treat multiple types of cancer. So what researchers found is that diabetic patients taking metformin actually have lower incidence of cancer. And the types of cancers would include prostate cancer, breast cancer, and colon cancer. And in fact, there's over 100 clinical trials that are actively going on right now to assess the benefit of metformin in cancer patients that are non-diabetic. So this is really impactful because, like I said, this is one drug repurposed to fight multiple types of cancer. Another interesting discovery about metformin is how it works. So of course we have a drug now that may be effective in cancer, but another information that we can really gain is how does this drug work? And if we understand how this drug works, then we can design new drugs to target cancer cells. So the second discovery is actually how it acts on cancer cells. It has both direct and indirect effects on cancer cells. The indirect effect that metformin has is really lowering glucose levels systemically. The direct effect it has on cancer cells is it stops the growth signals that are constantly activated in cancer cells. So as I mentioned, cancer cells have active growth signals, and the other observation is cancer cells actually are very, very hungry, and they take up a lot of glucose to sustain itself, enable for it to divide. So what we found out from metformin is it prevents cancer cells from taking up nutrients, and secondly, it prevents these growth signals that the cancer cells are responding to. So again, what is the impact? The impact is that now that we understand a little bit more about how cancers behave because of metformin, now we can design potentially new therapies to target cancer cells. So this is where we currently are, but where are we going? So far the examples that I gave to you are really serendipitous discoveries. You know, we found thalidomide happens to be a great anti-cancer drug. We found diabetic patients taking metformin have lower incidence of developing cancer. Well, now scientists are actually actively doing research to identify old drugs that can be used for cancer treatment. And you can see on the diagram on the right is actually a plate, and it has a lot of holes in it, and the holes in the plate would have different types of cancer cells. So researchers are designing experiments to discover anti-cancer activities in old drugs. So they're using it in a very automated fashion, using robotic equipment to screen existing drugs in the fight against cancer. So as I was mentioning, those little vials would contain cancer cells, and we can inject different types of drug into the vials and see how cancer cells behave. And of course, we're really interested in the drugs that kills cancer cells. So I just mentioned about doing high throughput screening techniques to find how old drugs can be used for cancer therapy. Well, this is actually ongoing right now, ongoing research happening in Toronto. So the researcher here that I'm showing, his name is Dr. Aaron Schimmer, and he's a medical oncologist at the Princess Margaret Cancer Center, and he is developing cancer drugs from old drugs. And in fact, the type of progress he has made so far is tremendous. He's actually advanced 3 drugs from his lab, so from cancer cells, all the way to clinical trials, so bringing it to the patients with leukemia and blood-related disorders. So just to summarize what I mentioned in this section about drug repurposing, the take-home message is that you can teach an old drug, or old dog, new tricks. You know, drug repurposing is another way of discovering cancer-fighting drugs. In addition, there are breakthrough cancer treatments using old drugs. And lastly, not only will there be additional treatment options, but we can really bring these drugs to the patient at a much faster rate. So just to close now, we want to remind you that the overall survival for cancer have improved tremendously from 20% in the early 90s all the way to 62% today, so 3 years ago, so obviously it has increased since then. You know, we're treating more cancers, we're treating cancer more successfully, and this is really due to early detection, better diagnosis, and what we've been discussing today, really about designing more effective therapy. So significant advances have been made for many major types of cancer, and this is largely because of the research that is happening here in Toronto and, of course, worldwide. But more work needs to be done, and that is why cancer research is so important. Cancer research is important because what it means is that fewer Canadians will face cancer, more Canadians can survive cancer, and really this would improve quality of life for Canadians during and after cancer. So of course we're unsure of what the future may bring, and what's important is that there's unlikely that there will be a single cure for cancer, but scientists like us are really working hard to make progress and make a difference. And hopefully you were able to understand some of the research that we're doing today and in the future. So at this point, I would just like to thank you so much for your time and listening to us today. And of course, we are more than happy to take any questions that you guys may have.
Okay, thank you so much for this presentation, Natalia, Sue, and Matthew. It's been really, really great. And now that we've heard from our researchers, we're going to go over some questions that you've been writing to us. So if you haven't had a chance yet, it's not too late. To submit a question, all you have to do is type it into the question box and then hit submit. And please note that our presenters will not be answering personal medical questions and cannot comment on personal diagnoses. So before we do jump into the questions, we have received several questions about funding these drugs that have been mentioned, and also asking about how people can afford them. So we agree that accessing medications and the high costs associated with them are an issue that the Canadian Cancer Society is very concerned about, and our advocacy department is really trying to help to improve this. So we do not have the answers for those particular questions, but we did want to let you know that it is an issue that we're working on. So the first question that I'd like to ask to our researchers today is it's going to be from a user who's typed in and asks, suppose cancer cells escape from the tumor. Can T cells find those cells, or does it have to be in a tumor to be attacked?
Okay, so I can address this question since it's kind of an immunotherapy question. And so what the person has asked is, when a metastasis occurs, which is when tumor cells from one tumor site move through the bloodstream and can attach themselves to other organs, and so lung cancer can move to the colon, or colon tumor cells can move to the heart, or things like this. If there is an immune response, can that immune response carry through to the new cell that has set up at a new location? And in theory, the answer is yes. If the tumor cell that has migrated has the same set of cell surface proteins, then the tumor cell, then the T cell will still be able to recognize that cell surface protein because the protein has not changed. However, there are differences in different environments. And so I mentioned that one of the ways that tumor cells escape detection by the immune system is by secreting chemicals that suppress immune system function. And so you can imagine that at a primary tumor site where a tumor has been for a long time, there might be a high concentration of these immunosuppressive chemicals because the tumor has been producing them for quite some time. If the tumor cell moves to a new site, there no longer is that buildup of immunosuppressive chemicals. And so, in theory, the immune system might even be better able to fight cancer cells that have metastasized and move to different areas. And so I just want to qualify what I'm saying. I don't know if any specific research has shown what I've just talked about, but based on my understanding of immunotherapy, in theory, the things that I've said are definitely possible.
Okay, thank you, Matt. So our next question is, in an article in Scientific American last year on cancer, one writer actually used the word resistance in describing the actions of cancer cells. Will you please give us more information on this?
So I can answer that because some of my research does focus on drug resistance. So I guess that paper, what they're talking about is some cancer patients that are on drugs may show effectiveness. So in terms of tumors might shrink initially, and this is great for the cancer patient, of course. But over the course of the treatment, the tumor may come back. And when the tumor comes back, this is what we describe as resistance to drug, so drug resistance. So there's a lot of active research around drug resistance, and resistance usually comes because of targeted therapy. Not always, but we usually see it in that, and that's what Natalia briefly mentioned during her talk. So one of the strategies that researchers are working tirelessly around is trying to discover and understand why these tumor cells become resistant to the drug. And of course, this is where combination therapies come in place, where we can, if we understand what drives resistance, for example, maybe they have a specific protein that is upregulated that helps them with the resistance. And if we understand that, then we could potentially use another drug to target that protein to use it in combination with the therapy previously. So in summary, I just want to say that drug resistance does happen, and we do see it. Not everybody will develop drug resistance. It really depends on the patient. But when that does happen, there's active research that is ongoing to understand the resistance so that we could still help the patients who are not responding to the previous drug.
Okay, thank you, Sue. So our next question, is chemotherapy a targeted therapy?
Okay, so chemotherapy is not a targeted therapy. Chemotherapy affects any cells that have a rapid cell division rate. So that's why when patients have chemotherapy treatments, they will develop gastrointestinal problems, they will lose their hair, because those cells, even though they're not cancerous, also divide very quickly. And so chemotherapy is not targeted in that any cell that will quickly divide in the body will be affected by this treatment.
I just want to add to that. So while chemotherapies don't target specific genes, chemotherapies can affect specific pathways in the cell. So every cell has many mechanisms to grow. And say there's 3 pathways, and all of them, when stimulated, tell a cell, I need to grow. What chemotherapy drugs try to do is block those cells from growing, blocking the cell growth pathways. So typically a chemotherapy drug might target one of these pathways, and it's not targeted because it affects every cell in your body. All the cells in your body are being told, do not grow. And this can have very serious side effects. But chemotherapy agents can affect only one of these growth pathways. So if you have a tumor and it's highly dependent on growth pathway 1, and we've given you now a chemotherapy drug that's specific to growth pathway 1, you might get a response and your tumor might shrink. However, the tumor cell can evolve to now use growth pathway 2 and growth pathway 3 through mutation. And so that is one of the mechanisms that tumor cells get resistance. And in the question that we previously answered, that is one of the ways that tumor cells can develop resistance to chemotherapy drugs.
And just to add to that, which is a note I find interesting, so there's current research trying to link the aspect of targeted therapy that makes it targeted to a specific type of cancer, for example, HER2, which I mentioned in my talk, and attach that little bit that makes it targeted to a chemotherapeutic drug. And so what they're trying to do is make targeted chemotherapy by having the chemotherapy only be delivered to cells that contain the receptors that they're targeted for. So that's another avenue that researchers are looking at.
Okay, wonderful. Thank you both, Natalia and Matt, for that one. The next question that we have is, it's more of a two-part question, but they ask, do cancer stem cells behave the same way as the cancer cells in the developed tumor? And how do viruses such as reovirus relate to immunology?
Okay, I can tackle the first part of the question. So it's a very good question, and it reveals a lot of understanding from the person who's asking the question, because this is kind of a new theory of how cancer exists in the body. So for those of you who are unfamiliar with what a cancer stem cell is, there's a theory that not all cells in the tumor are the same. There are only a subset of cells that are capable of forming a full tumor. So say you have a tumor and it's composed of millions of cells. Some of those cells, if you take them and put them into a new body, some of those cells will have no effect. Even though they're tumor cells, they're not capable of forming a full tumor on their own. Some of the other cells in that tumor, if you put them into a new body, those cells will be able to give rise to a full tumor. And those cells are known as cancer stem cells. So much like normal stem cells in the human body can give rise to all of the organs and all of the cells in a normal functional body, a cancer stem cell can give rise to all of the different types of cells in a tumor. And so cancer cells are very different from other tumor cells. One of the main differences is that cancer stem cells divide very slowly, whereas the cells in the bulk of the tumor divide very quickly. This is actually a big problem when it comes to resistance to chemotherapy drugs because, as I said before, a lot of chemotherapy drugs target the growth pathways of cells, the pathways that are telling tumor cells to grow really quickly. And so when you eliminate those growth pathways, the cells that are growing very quickly die very quickly because the pathway is no longer active. However, in the cancer stem cell, it is not addicted to that fast growth pathway. The cancer stem cell is growing very slowly. And so there's a theory out there that all resistance to chemotherapy and other types of therapy is because while the initial therapy kills the cells of the bulk tumor, the chemotherapy will kill most of the tumor cells. It will not affect the cancer stem cell. And because one of the properties of cancer stem cells is their ability to give rise to all of the different cells of the tumor, having cancer stem cells persist is very bad and will ultimately result in another development of cancer.
But there is hope. I just want to say that. That what Matt just mentioned really highlights the rationale in terms of combination therapy. So of course, the first question about what chemotherapy does, and Natalia and Matt touched about how chemotherapy are targeting the rapidly dividing cells. And now we have about a question about, how about these cancer stem cells? What about these guys? And yes, they are avoided from chemotherapy, but there are other types of therapy that does target cancer stem cells. An example would be what I just mentioned, so metformin. So I talked about metformin drug repurposing, how metformin is typically used as an it is used as an anti-diabetic drug, but one of the main features and one of the interesting features that researchers found about metformin is that it actually does kill off cancer stem cells. So that's a wow factor, and what that really means is we can actually use metformin potentially in combination with chemotherapy to really debulk and kill the whole population of tumor cells. And that's there is an ongoing clinical trials trying to assess the efficacy of that.
Okay. So the next question was about reoviruses, right? So does anyone?
I know a little bit about reoviruses.
Yeah. Okay.
So they're very interesting in that reoviruses are used, or we're trying to use them to treat tumors. because they are brought into the tumor by macrophages, so immune cells that are circulating in the blood. And once they come into the tumor, they will go inside of cells, but they will only be able to reproduce inside of cells that have a very specific mutation. And so in this way, it is a type of personalized medicine in that patients or cells that don't have this mutation won't benefit from the virus. So once the virus enters the cell, if the cell has this mutation in a gene called RAS, which is a very potent and known oncogene, the virus will replicate and will end up bursting the cell, and the cell will die. And so all those more copies of virus will now come into more cancer cells that, if they have this mutation, will lead to cell death. And so in this way, the virus goes in the whole body, but it only affects the cells that have this mutation and not non-cancer cells.
So it's basically a virus that targets specifically cancer cells and kills them. So we're using one of the properties of a virus, which is its specificity to detect certain cell types, and taking advantage of it and developing a virus that can target cancer cells.
So it's very lucky for us. And also RAS is a mutation that's found in a variety of different cancers, so it's not specific to just one type.
Okay, thank you. Thank you all for answering that one. So we have time for one more question. So we've talked a bit and asked questions about chemotherapy and asking if it's a targeted therapy. And we have another question asking if chemotherapy and drugs like tamoxifen are targeted therapies.
So I can answer a little bit of that. They are considered to be targeted therapies because tamoxifen is a drug that targets a specific receptor in breast tumors. So this is the estrogen receptor. And tamoxifen blocks the effect of this receptor. So cells that, or breast tumor cells that have this receptor, they overexpress it in the cell membrane. So
can you explain what the estrogen receptor actually
does?
So
the estrogen receptor is a receptor that will bind estrogen that is freely circulating in our bodies, and it will lead to processes of cell division and proliferation, which is normal in the breast during development, but not during adult life. And so, sorry, where was I? I lost my track. Right, so tamoxifen blocks the effect of the estrogen receptor, and these cells that are now addicted to the effect of estrogen receptor will not get the signal anymore, and they will die. So it's considered to be targeted in that only cells that overexpress the estrogen receptor benefit. If you have a tumor cell that doesn't have the receptor, there's nothing to block, and so it won't help at all.
Okay, thank you. Okay, so thank you so much for answering all those questions. We have had quite a few different ones come through, and I just want to let everyone know that if you have a question that hasn't been answered, or if you think of one over the next few days, you can always call our Cancer Information Service at the number listed on the screen. There are staff members there who can answer your question. And I would really like to thank all of our researchers once again for this wonderful presentation. And of course, I'd like to thank all of you as well for attending today's webcast. The webcast will be available for viewing in a few days, and you will receive an email with the information to view it. So keep your eyes to the community as well, because our next webcast will be on May the 26th, and we'll be covering the truths and myths of exercise in cancer. And please submit your survey about today's webcast too. This information will help us shape webcasts for the future, and we really want to create content that you're interested in learning more about. So thank you again for attending today's webcast, and a special thank you to Natalia, Matt, and Sue for presenting. I hope you learned something new and had your questions answered. And of course, I hope to see you back in the community as well, and I hope that you'll have a great rest of your day. Bye, everyone.